Welcome back to Paleo Talks. We're on episode 59, and today we're here with Hans Seuss. How you doing, Hans?
Fine. How are you?
We're doing really well. Excited to have you on the show.
Oh, thank you for inviting me.
You bet. And we're here with David and Chris as usual, and we have a few students that are hanging on here as well that are part of a paleoherpetology class here at ETSU, and so they may be showing up occasionally as well, but we're trying to keep them under control. And what we do first here is let David tell everyone, remind everyone about how the show works. Go ahead, David.
All right, just like normal, we are going to start off in just a second by introducing our guest and then going into our main presentation. The presentation is going to be the bulk of the program, but once the presentation is done, the rest of the program is Q&A with the audience. So when that time comes, feel free to submit questions for our guest in the comments of the Facebook video. And as always, if you can't leave a comment on Facebook for any reason, you can leave us a comment on our Gray Fossil site, Twitter or Instagram. And since we have a couple of extra people in the Zoom call today, if anyone on our call has a question they want to ask, go ahead and put it in the chat. I will be keeping an eye on all these different places, and we'll see if we can get Hans to answer as many questions as possible.
Thanks for managing all that, David. And we also have Chris Widga, as usual, with us today, and he keeps freezing up, so he's not just sitting really still.
I can sit very still, but yeah, today I may be freezing up a little bit, hopefully not too much.
All right, well, just as a reminder, we're coming to you from the Center of Excellence in Paleontology at East Tennessee State University, which oversees the Gray Fossil Site and Museum. And again, we're just thrilled to have Hans with us here today. And Hans, the way we start out actually with our introductions is we let you introduce yourself, and we want you to tell us all about how you got into paleontology in the first place and that sort of evolutionary track that you went on to get to where you're at today.
Yeah, my name is Hans Seuss. I'm a curator of vertebrate paleontology at the National Museum of Natural History, which is part of the Smithsonian here in Washington. I got interested in fossils very, very early on, sort of like around the age of 3. I was already interested in getting minerals and picking up fossils. Unfortunately, my home region in Germany was one of those Pleistocene deposits. There wasn't much. I found a few broken up pieces of mammoth bone, but that was about it. But then at age 4, the real sort of turning point in my life came about when my parents gave me a book on prehistoric life. It's written by this Czech team of Josef Augusta, who was a paleontologist, and he worked with this academic painter, Zdeněk Burian. And they really did the first really beautiful sort of artistic reconstructions of prehistoric life, so in the vein of Charles Knight, but in some ways far more diverse in terms of coverage. They would do Cambrian scenes, a lot of dinosaurs and so on. And I was just so impressed by the fact that there had been all these organisms once upon a time. I love living animals, but to have these extinct organisms that were stupendous in size, had weird shapes, lived in really weird places, that just sort of fascinated me. So I told my parents at the ripe old age of 4 that I was going to be a paleontologist. Now, in my family, other than one chemist, we hadn't had any scientists at all. They're all in law. My dad was in banking, or some of my uncles were medical doctors. And so my parents hoped that, you know, at some point I would sort of finally see the light and embark on a more promising career than paleontology. But I stayed with it, and in my teens, I joined a fossil hunting club that traveled all over Europe. There were people like policemen, lab technicians, and so on from various places. And we all had this shared love of fossils. So we're mostly looking for invertebrates. But I remember finding my first fossil vertebrate, which was a partial Oligocene sea cow skeleton. And that was very exciting. In the same pit, there were also lots of shark teeth. And so this really sort of set me on my future course. Did my undergraduate work in Germany, and then I got an exchange fellowship to Canada. So I went to the University of Alberta in Edmonton. And I chose Alberta because that was the big dinosaur hunting ground. And this was before the days of Phil Curry and things like that. In fact, Phil just arrived there when I was leaving to go to my next academic station, which was Harvard University, where I got my PhD in vertebrate paleontology. And then I did postdocs at McGill and at the Smithsonian. And then my first job was at the Royal Ontario Museum in Toronto, who had a joint appointment as a curator of vertebrate paleontology and a professor of zoology at the University of Toronto. And then I had a sort of brief stint at the Carnegie Museum of Natural History as the assistant director for science and research. And then I came back to the National Museum of Natural History, first as a senior administrator. And then after a couple of years, I finally had a chance to go back to research, and I did that. So.
When did you start thinking about turtles and other early reptiles as areas of your focus?
Well, I've always been interested in modern turtles, but I had never actually found much of turtle material, some Cretaceous turtle material when I was working out in the Western interior and then later in Central Asia. But the Triassic turtle that I'm talking about today was a very interesting chance discovery. There's a new deposit in southern Germany. It's Middle Triassic in age, so about 240 million years old. And it had yielded a lot of fossil reptiles. And I was going through the collections, and I had started a number of projects with my colleague Rainer Schoch, who's the curator for fossil amphibians and reptiles there. And I came across these strange little bones. And I said, Rainer, this is a stem turtle. And he goes like, really? No, it can't be. And I said, all right, wait. So we spent a whole afternoon looking at every bone that we had. There were a few sort of associated postcrania and so on. And at the end of the day, we just went out and drank a lot of beer and celebrated the fact that we had indeed found something that looked like a stem turtle. However, a lot of my colleagues said, no, it doesn't have a real shell, so it can't be a turtle, the sort of typological thinking that sometimes holds us back. But as we looked at the bones, more and more features emerged that it was indeed a stem turtle. So that's how I came to turtles. I always liked them. I never had a chance to find anything like Cretaceous or Cenozoic that I could work on. I'm very broadly interested. As Lou Jacobs once said, there's no fossil I don't like. So, so I was very happy to, finally get a chance to get into turtles by means of a stem turtle, at least. So,
no, it's a really exciting specimen, and we're ready to hear more about it. So if you want to go ahead and share your screen.
Yeah, thank you. See that?
I don't see it yet.
Okay.
David, make sure that he can share his screen.
Yep, should be able to.
Yeah. Good now?
Yes.
There we go.
Okay, good. Okay.
All right, looks good.
Today I want to talk about one of the oldest issues in comparative anatomy, namely how turtles got their shells. This is a question that had preoccupied naturalists for literally millennia. Aristotle was already pondering the issue. Although at that time, the ancient Greeks had a great mythology, namely that there was this nymph who was invited to the wedding of Zeus and Hera, but she was kind of, I don't know whether it took her too long to put her makeup on or whether it took her too long to just leave the house and get to the wedding. But in any case, Zeus was absolutely incensed that this nymph had not come to his wedding. So he sent one of his messengers to her and said, like, what's the deal here? And the woman said, oh, well, I just like being at home. So as a punishment, he instructed this messenger to turn her and her house into one creature. So this creature, this nymph named Chelone, became then the first stem turtle. In the 19th century, Georges Cuvier pondered the issue again because he could really not work out how an animal that had basically integrated its skeleton into armor could have come about. So he started, for instance, thinking that perhaps the plastron, the ventral half of the shell, was the equivalent of a sternum and so on. But again, it just didn't really work out. But then with the 19th century comparative anatomy, particularly in Germany, people sort of started making all kinds of interesting observations about how turtle shells developed, but somehow it wasn't sort of put all in an overall frame. And over time, when paleontologists were looking at the issue by themselves, it was really sort of something that puzzled them, and they sort of said, well, you know, how would we even recognize what a turtle precursor would look like? You know, you can't really imagine going from nothing to a fully developed shell. And also the same argument that a lot of people had in those days, late 19th, early 20th century, namely, how do you get a complex structure that wasn't there before? Are the individual steps potentially selectively advantageous, or is this just something like later on Goldschmidt called hopeful monsters, that literally the first turtle with a full shell hatched from another kind of reptilian egg? So in recent years, people really got interested in the question again, and it was in part because embryologists had suddenly become fashionable again, and with the new methods that we now have with radioactive markers, trying to link particular structures to particular genes and domains, it's actually much easier now to understand how the shell develops in modern turtles. And so we had a little bit of basically a blueprint of what we could be potentially looking for. Before I get into further details, I want to acknowledge two of my close colleagues and fighters in the trenches of turtle evolution. Rainer Schoch, who I mentioned earlier, I worked with on Triassic reptiles from southern Germany, and my colleague Thorsten Scheier, who teaches at the University of Zurich in Switzerland, who is one of the world's best paleohistologists, and it turned out to be very important for our project. But Thorsten, like me, is also broadly interested in Triassic reptiles. When we look at turtle shells, there's an amazing amount of variation, even though there's sort of one general body plan, namely that basically the entire trunk is enclosed in bone. But when we look even at the 355 plus species that are alive today, there's a great deal of variation. At the top left is one of those great Galapagos tortoises, which is sort of the ur form of the turtle, as most people are concerned, a complete shell with a carapace and a plastron. Then below it is a turtle that has a fully developed shell, except between the individual bony elements, there are huge spaces so that the whole turtle shell is very flexible. This is a pancake turtle from Africa, Malacocheirus, and this animal can literally wedge itself into crevices in the rock and basically becomes impervious to attempts to prey on it. Then on the top right, we have a soft-shell turtle, the Trionyx, which has greatly reduced its carapace and to some extent the plastron, and has instead replaced them with tough leathery skin. And then below we have a marine turtle, and marine turtles we can trace back to the late Cretaceous, and we can see over time how the shell becomes progressively reduced up to the point that we have in the great leatherback turtles, where they're basically just little ossicles in a very tough epidermis, and even the plastron is reduced to just a very thin ring of bone. And this seems to be sufficient for this animal, because the problem, of course, for a marine turtle is you don't want to have any extra ballast when you're swimming around. And some of marine turtles travel for thousands of miles to get to breeding grounds or to get to feeding grounds. So clearly there's a premium to get rid again of this turtle shell. Now the turtle shell is basically a two-material system. We have a bony shell, here shown in the sort of light yellow, which is composed of bony plates. And these are overlain by keratinous scutes, the kind of material that we have in our hair and fingernails. And the keratinous scutes are offset relative to the bony elements, which is very handy for paleontologists because the keratinous scutes sort of leave little grooves where they attach to the bone. And so even on a Jurassic turtle, we can figure out what the keratinous scute configuration looked like. But the advantage of this sort of two-phase system is that it really mechanically strengthens the shell. People have done biomechanical experience on shells and found out that if you have just a bone shell, it has much less compressive strength than if you have the keratinous material on top. So this is really a very important aspect. And so we have this both on the back as the carapace and then on the ventral side as the plastron. Now, the most obvious function for a turtle shell is presumably a protective function. Here you see a tortoise being tossed around by a Nile crocodile. Remarkably, this particular tortoise got away because the crocodile could not position it at the back of the jaws, where it would have had bite force sufficient to actually crack the shell. So it just wandered off. And this must have happened millions of years at a time. Here's a late Cretaceous soft-shell tortoise from Alberta, and you can see here in the circled area, a broken area where clearly probably Lydiosuchus, which was a common alligator relative at the time, tried to bite it. It pushed the bone in a little bit, but it healed in place, and apparently the turtle led a happy life for quite a long time afterwards because of the degree to which this injury has been healed. And we see the same thing in a lot of modern turtles. Often you find turtles like here in the southeast with spectacular shell damage, but they soldier on, and reptiles have a really powerful immune system, as anyone who has kept reptiles knows. And so they just mosey on and eventually redevelop the broken areas. Now, the problem with looking at the question of the development of the shell for paleontologists was that for a long time, the very earliest turtles, or we would call them now stem turtles, because we now restrict the term turtle to a much smaller group of animals, but they came from the late Triassic, about 250 million years old, first in Germany, but since similar specimens have been found in South America, in Thailand, in other parts of Asia. And so, but when you look at them, they're already perfectly good turtles. There are slight differences in the arrangement of the bony plates in the Carapace and Plastron, but this is a turtle, so this is not going to lead us any further. And as recently as 2018, I saw a quote in a book by one of our senior colleagues saying that, basically we still have no idea where turtles came from because the earliest turtles are already turtles. So when I talk about turtles here, I use the term broadly in the traditional sense. Nowadays in phylogenetic classification, turtles is restricted to the last common ancestor of Pleurodires and Cryptodires, two major recent groups. And so a lot of animals that are more basal than this node are called stem turtles.
Hans?
Yeah.
Now those turtles do have teeth as well as other early ones.
Polkadot, the one on the right, does. And they're tiny teeth, and they're mostly on the palate. We don't know yet what the skull of Proterocercis looks like, but we have other Triassic turtles, now stem turtles, that actually have teeth, and some have already gotten rid of the teeth. And I'll point this out as we go along. Now, the interesting thing about turtle shells also is that they're completely unlike similar structures in other vertebrates. On the left here, you have a Placodont, a Triassic marine reptile, and this is an extreme form of Placodont that has entire body enclosed in bony plates. The arrangement is quite different from turtles, but the significant point is here when you look at the lower right, the lower left, you see this cross-section of the trunk of this animal, and the axial skeleton, the vertebral column and ribs, are not firmly attached to the bony plates. They're actually quite separate, and there presumably was only ligamentous connection between them. Among mammals, when you look at the giant armadillos like Glyptotherium, Glyptodon, and so on, they have a massive carapace. But again, as you can see in the picture below, the skeleton is entirely separate and not fused to those bony elements. And that was a problem because when you look at a turtle shell, it had been suggested early on, so this is a carapace, and you're looking at it from below, you see the vertebral column. Between two vertebrae, you always can see a little rib going off. And this was a structure that is really without parallel among known vertebrates, at least. But for a long time, people thought, well, maybe given that these other animals all have osteoderms, these bony plates, and ribs that are separate, maybe what happened in turtle evolution was what Olivier Riepel has called the Polkadot scenario. You started out with an early reptile that had lots of bony plates embedded in its epidermis and dermis. Then you got something where these elements were sort of fusing more and more together. And then you finally got to turtles. And then once you had these big plates, they started fusing to the ribs. And this was like for many years, well into the 2010s, the way that people looked at this. And there was also a fossil reptile, a group of fossil reptiles that seemed to fit this. These were pariasaurs, a group of, they were really the first large reptilian herbivores in Earth history from the middle and late Permian. And some of the forms have extremely heavy armor, and in some areas here over the shoulder and over the pelvic girdle, you can already see that these plates are sort of coming together. In these cases, they're actually sutured together. This is a form Anthodon, which is one of the most derived pariasaurs from the middle and late Permian. But the problem was that it didn't answer some other problems that we had with the idea of developing a turtle shell. When you look at the standard amniote, you have the shoulder blade outside the rib cage. Yet if you cut through a turtle skeleton, you see that the shoulder blade is actually inside the shell and sort of close to the vertebral column. So basically, in other words, it's basically inside the rib cage rather than outside as in other amniotes. And this was a puzzlement that people, I remember papers by DMS Watson from the 19-teens. And so they suggested, well, maybe the scapula or the entire shoulder girdle sort of moved back within the rib cage during ontogeny at some point. But then in the 1920s, when people first started looking at the question in more detail, it turned out that the ribs were actually growing out over the shoulder girdle. This here, just to give you an idea of the scale, this is 1/10 of an inch across. It's an embryo of a sulcate tortoise from Africa. And you can see the ribs very nicely. They're still so short. They're already getting a little bit expanded. And then you see that the scapula is already covered by the ribs. So literally the ribs grow over the shoulder girdle. And the same thing happens in the pelvic region. Now, when you look at this developmentally, it turns out that there's a fundamental difference in the way turtles grow their ribs from all other amniotes. In most amniotes, the ribs grow from the vertebral column downwards and sideways. And you can see here the relationship to the shoulder girdle. The shoulder girdle is the structure over to the side. You see the arm. And you can see the serratus anterior muscle attaching and basically suspending the shoulder girdle from the rib cage. In the turtles on the right, you see that the rib does not grow that way. In fact, the rib grows just straight out to the side. And this is induced by an area called the carpacial rib zone that is actually a zone of mesenchyme that triggers the growth of the rib, the stem cells for the rib to sort of go out to the side. And you see that the serratus muscle here now actually sort of projects inward because it's now trapped along with the shoulder girdle inside the rib that grows over it. And this was demonstrated by actually sort of selective dyes, and now we understand even the genetic base. We can specify individual genes that are responsible for the development of this carapacial ridge. Not only does the carapacial ridge allow the ribs to grow outside rather than outside and down, but it also is responsible for the fact that in many turtles you sort of have this sort of fan-shaped arrangement of the ribs, and this is also controlled by the carapacial ridge. So much for the embryology. No, there's a little bit more. Here's how the ribs actually develop. Deep in the, you see the dermis here, DM, you see these condensations of cartilage cells, and then you get the sleeve of perichondral bone enclosing the cartilage. And then at a certain point, this sleeve develops these little flanges and they progressively grow out to the side. And this is what forms the actual costals of turtles. Now, in terms of stem turtles, this is basically all this interesting embryology got us to look back at stem turtles. And my friend Tyler Lyson sort of looked at a middle Permian reptile from South Africa called Eunotosaurus africanus. When it was first described in 1892, the researcher describing it already thought, this looks a little bit like a turtle. And then in 1914, DMS Watson, a very prominent early reptile paleontologist, actually said, this gives us a good idea of what he called archichelone, the sort of original body plan of turtles looked like. It had a foreshortened trunk region, which turtles have relative to more primitive amniotes. It has these broad ribs. And he also noticed that it had a fairly restricted number of neck vertebrae. And all in all, this seemed like a really good model, anatomical model of what a turtle precursor could look like. This idea was popular for a few years, but then researchers said, well, it doesn't really look like a turtle, so it has to be convergent. And that was sort of the received wisdom that you find in all of the textbooks until Mike Benton's recent textbook, that basically, yeah, kind of looks like a turtle, but isn't the right thing. Here's the expanded ribs again. So in comes this little turtle that we had from southern Germany. It's from the late Middle Triassic, about 240 million years old. And here's much of a skeleton. You can see a very long tail trailing off to the left. And then in the center, you see these very expanded ribs, actually more expanded than in Eunotosaurus. And the other interesting thing is that they have distinct sculpturing on them. There are other features that probably go too much detail here, but for instance, the pubic bone has this very prominent lateral tubercle that all turtle pubes have, and lots of other little sort of details that I don't want to go into here because they're not pertinent to the main story. So here are the ribs in more detail. You can see they have a central shaft, so the original rib, and then just like in Eunotosaurus, there's a flange growing out in front and behind. When you do a histological section, you see a central cavity, which is where the rib cartilage would have been, and you see these flanges in laminae sort of growing out forward and backward. And basically in Pepperquil, these ribs already slightly overlapped. And that had profound implications because it led to a reorganization of the intercostal musculature. And more importantly, once you expand the ribs to a certain extent, you are no longer able to respire by the standard costal respiration that most amniotes use. So this is something that certainly is very turtle-like. Now, there was one bone that we had a lot of problems with, and that's the nuchal bone. It's this big unpaired bone at the front of the carapace, here shown in red. And it took a mostly comparative anatomical approach to figure out what it was. It is the clithrum, the precursor for it is the clithrum in primitive tetrapods. Most modern tetrapods have lost the clithrum altogether, but if you look at Permian, this is a dimetrodon shoulder girdle, you see this very prominent bone. You have the interclavicle at the bottom, the clavicle in front, it's broad spade-shaped thing, and then above it, the clithrum. And the reason we think that the clithrum and the nuchal are the same is that the nuchal actually starts from two embryonic precursors, and it has exactly the same relationships to two of the major muscles, the trapezius and sternocleidomastoideus muscle, as it does in, say, lizards and so on, and forms that at least have still rudiments of the clithrum. So even though we don't have a clear intermediate stage here, we can see that this is likely the homologue of the clithrum in non-turtle vertebrates. Now, why would one expand ribs? My former doctoral advisor, Farish Jenkins, studied edentates in the early 70s to figure out a similar question with some non-mammalian cynodonts that have broadly expanded lumbar ribs. And one of the ideas that he came up with is that it really allows you to stabilize the back because you basically create a rigid structure, particularly in a reptile that has very much lateral and medial flexion. It's very important to do that. He used as an example here, this partial skeleton is from a silky anteater, Cyclopes. And Cyclopes makes its back really rigid when it reaches from branch to branch and basically needs a very stable platform so that it can optimally employ its forelimbs. Now, for Eunotosaurus, we have no evidence that it was climbing around in trees, but we do have evidence that it was likely a fossorial animal. It has enormous claws on its fore and hind limbs, a massive olecranon process on the ulna. And so for an animal that's digging, it makes sense to get yourself a really rigid back, because if you use the forelimb as your primary organ for excavation, you want to have a stable trunk region so you don't waste effort by constantly counterbalancing against the lateral flexion, which you would do if you had a normal reptilian postcranial skeleton. So the fact that Eunotosaurus most likely was a burrowing animal may have triggered this initial development of expanded ribs. And then once you were on the way to that, many of the other modifications of the carapace fall in place. Now I come to the second part of the turtle shell, the plastron, the belly half of the shell. And this is the first record of a complete plastron. It's again in a late Triassic stem turtle, a little bit younger than Pepperquil, called Odontochelys. It's named Odontochelys because it still has teeth, marginal teeth, unlike most later stem turtles. But it's remarkable that it has a fully developed plastron. You can see these sort of little projections all over the place. But interestingly, you look, the ribs are still free. So there's no carapace. So the plastron actually developed before the carapace. There had been previously some embryological indications for this, but this was beautiful confirmation that this was indeed the case in the fossil record. Now, the plastron has been long suggested to be somehow related to a structure in other reptiles called Gastralia. Among modern reptiles, only crocodilians and the tuatara have these little things that are also known as abdominal ribs and so on. I find that a bad term because it implies that there's some homology to the real ribs, because these structures actually ossify in a very different way. There are structures that ossify within the rectus abdominis muscle of these animals, and they're dermal structures. When you look at turtles, in this, again, this example from the sulcate tortoise, you can see these two bundles of bony spiculae, and then you see the structure in the center, which has three parts, and so basically sort of T-shaped structure. It had been suggested by some recent work on the Gastralia in alligator that they were controlled by the same gene as these bony spicules in tortoises. In fact, you can actually sort of, there's a differential stain that only stains Gastralia and these bony spicules in turtle embryos. So there's a good case to be made just based on comparative anatomy that these might be homologous. Well, Pepperquil, our little stem turtle, brought beautiful confirmation of this. On the left, you see a bundle of these very massive Gastralia, and on the right, you can actually see examples where Gastralia are already fusing together, very much as what you see in the developing plastron of modern turtle embryos. So this gave sort of further credence to the fact that Gastralia and at least the major plastral elements are homologous structures. Here's a more derived stem turtle that was discovered after we had already discovered our animal, called Yowwrinkle. This turtle is interesting because it has already lost most of its teeth and has developed a nice beak over much of its snout. But the postcranial skeleton is still very basic. You still have broad ribs without a carapace, and you have these very thin structures here that are still the Gastralia, sort of more similar to condition of what you see in Pepperquil, and sort of very different from what you see in Odontocheilus, which is just from a geological horizon just above this one. So this must have happened very quickly. Now, part of the plastron is this T-shaped structure that I showed earlier. And when it had long been suggested that this T-shaped structure would be somehow homologous to parts of the shoulder girdle, particularly the ventral shoulder girdle. Here are the shoulder girdle bones of Pepperquil. It has already a very sort of tall vertical scapula, a little acromial process here. But what is interesting here is the interclavicle, which is this cross-shaped structure, and then the clavicle here with a broad end that overlaps this part of the interclavicle. And this leads back to something that people have pondered based on embryology. So here in a plastron of a modern tortoise, you see these two paired green elements. They're the so-called Epiplastra, and then you have the entoplastron in purple in the middle. When you look at the development of these structures, you see that the Epiplastron already early on sort of develops as paired, and you have this T-shaped entoplastron. And then at the bottom is an example of an early reptile, and it has these clavicles and the interclavicle in exactly the same position. So when we look again at what we see in Pepperquil, it has the same condition that this snapping turtle embryo at the top on the right side has. Two clavicles and then the interclavicle, and the next step is simply to fuse, to expand these into plates and make them the front of the plastron. So here's a reconstruction of the skeleton of Pepperquil. We now have well over a dozen partial skeletons and many isolated elements. In red, I'm showing you the broadly expanded ribs, and then in green, this Gastralia basket, which eventually will become the plastron. So we can develop this beautiful morphological transition series, starting here with Pepperquil. And Pepperquil shows already a number of the sort of traits like the Gastralia apparatus, the broad ribs, the foreshortening of the trunk and neck. Eunotosaurus has those features as well, but it has also some additional features like the lateral pubic tubercle that I mentioned, features of the humerus and femur, and so on. Then you have Yowwrinkle, which is basically the Pepperquil on steroids. Pepperquil is about a foot long, a little bit more maybe, but Yowwrinkle is between 6 and 7 feet long. So it's basically sort of a souped up version of Pepperquil, but it has some even more derived characters like the beak that it already has, great reduction of teeth, and so on. Then you get to forms like Odontocheilus, where you still have no real carapace, just broad ribs, but you have a plastron. And also Odontocheilus has gotten rid of a lot of other sort of basal features of the shoulder girdle and pelvis. And really the only thing that changes is that the skull becomes completely closed up and there's further reduction of the dentition. And then finally we get to Proganocheilus, where the whole paleontological story started out, where you have a fully formed carapace and plastron. The limbs are already completely enclosed in those, the proximal portion of the limbs. The neck and tail in this particular form are also armored, which most later turtles get rid of this extra armor. And most importantly, the skull has no temporal openings, which for a long time paleontologists argued was a primitive condition, because for many years it was thought that amniotes primitively had no temporal openings. But we now know, because Pepperquil and Eunotosaurus, at least as a young animal, have 2 temporal openings, that the turtles came from diapsid ancestors, and the turtle skull lately basically got consolidated. It has been suggested that this had something to do with since they have a very short neck, the powerful neck musculature that is exerting strong tensile forces on the back of the skull, and that this may have led to turtles closing up the temporal region. So turtles arising about the same time that we have also dinosaurs arising were quite a success story in their day. They were quite diverse in the early Cenozoic and in the Mesozoic, and survived non-avian dinosaurs by over 66 million years, and today are still a moderately diverse group, not as diverse as squamates, but still they obviously have a body plan that allowed them to adapt to various ecological circumstances. And actually, the more we learn about the physiology of turtles, interesting facts like, for instance, they can respire through their cloaca and things like that, we can see how in the KT boundary cataclysm, these animals could have survived, particularly the aquatic forms or the burrowing forms, whereas the larger non-avian dinosaurs would fall victim to this great catastrophe. So I hope that what I have tried to show you here is that there's really sort of nice consilience of data from embryology, comparative anatomy, and paleontology. And it's sort of one of the things that I now always point out to my neontologist colleagues, namely saying that paleontology has real data to offer. Some of you may be aware of the literature, particularly in the early 80s, when people said, well, fossils really don't tell us anything about evolutionary change. But no, they do tell us a lot, because they preserve the very stages that we often develop as hypotheses from developmental and comparative anatomical data. So thank you for your attention.
Thank you, Hans. I want to remind the audience to send in your questions. And if you're on here with us on Zoom, go ahead and type your questions into chat. You know, it's amazing to me, Hans, how, you know, as we come up with these extraordinary fossil discoveries like what you're talking about today, that it does take a while for it to trickle back into neontology. And so the textbooks that are coming out still tell, in many cases, the old story. So how do we get...
The turtle situation is actually better than most, because a number of researchers, particularly Japanese researchers who had done some really cutting edge embryological work, and also here in the United States, Scott Gilbert at Swarthmore College immediately jumped on the fossil evidence. And this is now very much part of the ongoing discussions about turtle shell development. There's still some issues to be ironed out. For instance, it turns out that soft shell turtles have some very odd things happening during their ontogeny. The basic pattern is the same, but there are these variations, and herpetologists and embryologists are very interested to account for these. And you know, so this has not been answered yet by either neontology or paleontology.
And it's also extraordinary how far back some of these groups go, like the Trionychids. You know, it's
Yeah.
from such an early time they had diverged and are so
Mhm.
distinctly different.
Yeah. Now, and where even there's now slowly a sort of record of fossil turtles coming out of the first half of the Jurassic, because basically modern turtles go back to like the late Middle Jurassic and late Jurassic, and anything before that are really stem turtles in modern parlance. But it's really interesting, some of the, like for instance, I'm aware, and unfortunately it hasn't been published, and I don't know when it will be published because it's in private hand, but I know of a marine like earliest Jurassic turtle from northern Germany.
Hmm.
So they were already marine at that point, so far earlier than anyone had ever assumed, because previously the earliest marine turtles were from the late Jurassic, where they're already quite diverse. So people had long expected that they would have a longer trail back into history, but the middle Jurassic is still very poorly documented around the world. But so there's quite a bit of evidence that's from the Cayenta Formation. There's this animal called Cayenta kelis, which was clearly an aquatically adapted stem turtle. It had a very flat shell, and it has various other, like the limb proportions and so on, are consistent with those of freshwater turtles today. So even very early on, there was already quite a bit of diversity of these groups, but it again sort of highlights, people talk about dinosaurs and other big things, but these sort of smaller animals haven't gotten the same attention. But if you actually look at the fossil records and look at it carefully, not just with these big things in mind, there's a lot of evidence out there that has yet to be interpreted. And like in my own work in the Triassic, even here in the United States, I'm always amazed how many of these animals clearly show anatomical features that link them with much later groups, and would, if phylogenetic analysis can actually sort of confirm this, would really drag some of the lines like way back. Squamates being a great example. Generally, the earliest known squamates are from the Middle Jurassic, but there's increasingly now material from the Triassic that shows that squamates have a very, very long history, and it may even predate the Permian-Triassic boundary.
I have a question, Hans.
Yeah.
You know, it really strikes me that these animals are just over-engineered. I mean, from early on, they've got this really robust shell. The disjunct timing between the evolution of the plastron and the carapace is really intriguing. They can breathe out their cloaca. So, you know, they're basically over-engineered to, you know, basically survive something. You know, we know that like crocodiliforms were munching on these things. Is there some sort of something that's going on in the Triassic that is really kind of pushing the evolution of this overbuilt, over-engineered animal?
Well, it could be. I always sort of, having now worked in most parts of the Triassic, I think of the Triassic as the vertebrate equivalent, or at least land vertebrate equivalent of the Cambrian explosion. You get this myriads of forms, many of which don't survive the Triassic. In fact, the Triassic is sort of, in fact, I saw there's a page on Facebook now called WTF Triassic, because the Triassic is generating so many utterly bizarre animals. And so out of this came a number of groups that really have a very wide range of adaptability. Crocodilians, one example, you know, in their stem forms, and turtles certainly are another example. But in terms of over-engineering, I don't think any of them are over-engineered, because still the old Wolff's law applies that bone is only deposited where it is mechanically necessary. So I think they're just, you know, just really well-adapted little animals that can do a lot of different things. And
phytosaurs would have been playing a role in this too, I
Oh,
suspect.
absolutely. Yeah, if a phytosaur gotten hold of one of those things, of course, some of those phytosaurs were much bigger than any of the modern crocodilians. So it probably just would have swallowed the thing and not even given it a second thought. And we do have, like petrified forests, we do have turtle remains, so stem turtle remains.
All right, David?
Yes, we have started getting questions from the audience. So as a reminder, if you have questions, go ahead, put them in the Facebook comments, or send us a message on Gray Fossil site, Twitter or Instagram. I'm going to start with a question from Jenny. Are there other species besides Proganachilles that show the evolution towards toothlessness in turtles?
Well, the first turtle that has virtually no teeth left is Yowwrinkle, the one that still has a very primitive carapace and plastron. And apparently, the presence of teeth in Proganachilles is sort of basically a retention of a primitive feature that then is lost. I don't think anybody has done any kind of experiments yet embryologically to see whether you could induce tooth formation in modern turtles, like they have done for chicken successfully, where you can actually re-engineer at least tooth buds in chickens. There seems to be some kind of genetic trade-off. If you get a beak, you can no longer develop teeth. This is just sort of the pathways that are needed for either some in conflict. It's the same thing. For instance, turtles have a plastron, but turtles are the only reptiles that don't have a sternum. And it has actually been demonstrated by a study just a couple of years ago that the precursor cells that are necessary for the sternum are the same ones that are necessary for the plastron, particularly for the anterior part of the plastron. So apparently, once you opt for a plastron, you can't have a sternum. And there are other sort of interesting morphogenetic trade-offs. And it has been suggested, particularly by the work of Laurent Viriot in France, that once you go down the road of beaks, you can't get teeth as well. So somehow the tooth development is suppressed in favor of a beak.
Very interesting. Well, speaking of plastrons, we have a question about plastrons. This is from Heather, who asks, how do hinged plastrons come into play? I know that box turtles can enclose themselves with their hinged plastrons. Do we know where that comes from?
No, this is actually unique to that particular lineage. Now, given that these sort of the plastron develops basically in segmentation ways, like for instance, when you saw the little bony spicules at the back of the plastron, there is a zone in between, and you could easily see a developmental pathway where that zone doesn't actually fuse up, and then you can actually fold the plastron in the way that Kinosternids do. And even the earliest Kinosternids already have that division, so, and none of their sister taxa do. So apparently it's probably a very simple switching on or off of a particular gene that controls that.
Very interesting. Here's another question. This is from one of our students, Sam. Of course, when you talk about ancient turtles, you have to expect that someone will bring up giant ancient turtles.
Yep.
So Sam has asked, would Stupendomes and Megalocheles be inertial endotherms like leatherback sea turtles?
It is possible, although both of them are basically continental animals, where the challenges in terms of ambient temperature are less than in the ocean. Like, the leatherback turtle can swim into temperatures that are just below freezing and do fine because it can actually generate internal heat through muscular activity. Plus, it has very good subcutaneous insulation. Whereas Stupendomes was living, and Stupendomes and also these new giant turtles from Colombia, from the Paleocene, were living in freshwater environments in very warm environments. So they wouldn't really have had any kind of issue in terms of having to raise their body temperature. Megalocheles is a slightly different story because this is a continental animal in the forelands of the rising Himalayas, where you would have had at best temperate weather condition. So it's quite possible that Megalocheles would use inertial homeothermy to control its body temperature. Certainly was big enough.
Excellent. Here is another question from Jenny, who asks, in this case, about the origins of turtles. What is your take on turtle origins in regards to where they fit? Are they a sister group of archosaurs? Where do they fall on the reptile family tree?
Well, according to our phylogenetic analysis, and I'm pleased to say Tyler Leeson and his colleagues found similar results, they are neither related to lepidosauromorphs nor to archosauromorphs, but that they're actually a separate group, even outside Sauria, as they're now called basically the modern diapsids. Now, the interesting thing is that among people who studied this problem from the neontological end, we have no evidence whatsoever that turtles have any shared derived features with archosaurs morphologically. When you look at the genomes or even at individuals' sequences, you don't have any problem relating turtles to archosaurs. Also, if you use a different approach to reading the sequences, namely that you take the sequences and just translate them into genes, then if you do a phylogenetic analysis, and they've done that both with Bayesian and parsimony, it actually turns out that you can either have turtles as a sister group to archosaurs, specifically crocodilians, or as a sister group to squamates, or as a sister group to neither, like maybe sauria in general. Now, there are a few morphological features that turtles have in common with squamates. There's sort of fine details of muscle attachment and so on. So not something where you would go like, oh, wow, yeah, that's the kind of thing that I really see links them together. Now, if you look at the fossil record, we now know that turtles are diapsids. So that part has been confirmed by the fossil record. But whether they have any connections with archosauromorphs or lepidosauromorphs is still very uncertain. There have been some phylogenetic analyses that found turtles closest to lepidosaurs and sauropterygians, the marine reptiles, but we couldn't confirm that in our more extensive phylogenetic analysis. So that's still a big open question. Why do the molecular data tell us one thing and the morphological data another thing? So, because the molecular signal is really strong, so that is very disturbing. Is this a case of long branch attraction, or what else is coming into play there? That's really sort of a thing that sort of keeps some of us still awake at night.
All right, we have one from Davis Gunnan, another one of our graduate students. When examined histologically, what types of bone growth occur in the carapace or plastron? Is there much remodeling throughout life?
There's quite a bit of remodeling in the carapace, but not to the same extent that you see in the limb bones and even in the ribs, the free ribs of other reptiles. So it's a relatively stable system. Although there is some remodeling, for instance, the shell is being used by turtles. For instance, female turtles, when they produce eggshell, will take calcium out of the shell. So you will sort of but nobody has ever sort of shown whether there's the equivalent of what you have in birds with medullary bone. When a female bird's producing eggshells, it does things to the histological makeup of the long bones. So that hasn't been looked at yet for turtles.
All right, we've got one more from the audience here on Facebook. This is from Nick, who asks, do you expect to find animals like Eunotosaurus in the early Triassic?
Yes. Definitely. The problem is that we have very few early Triassic occurrences that are A, safely dated as early Triassic, and B, that have yielded much in terms of sort of smaller vertebrates. You get these highly biased faunas, like for instance, South Africa, the Lystrosaurus assemblage zone, which is 90% Lystrosaurus. I'm sure there are little things were around, but there's very little of a record so far on that. But I'm convinced that as more and more sort of small vertebrates are found, there are now discoveries of small vertebrates, for instance, in the early Triassic of Poland. There's some Russian occurrences that haven't been published yet. So there's more and more of this material coming up. So I wouldn't be surprised if someday soon we would have something like a Eunotosaurus, or perhaps something even more derived in those bones. The problem is that a lot of these small vertebrate faunas are all isolated bones. And, even if you have a good, lively fantasy, it's sometimes very difficult to put these things back together. In fact, there were, like, for instance, for the Polish deposits, people have found very stamped squamates, but a lot of people criticizing them because they're basically putting different skull bones together. I think they have done so in an excellent and plausible way, but some people are just not very comfortable with that, unless you have something articulated there. Little skeptical. That's a general problem with studying small vertebrates, that they're often little bits and pieces, and you really have to know your anatomy, and then you have to really try hard to convince other people of the validity of your arguments.
All right. And then again, speaking about that weird anatomy, one more question from the audience as we get towards the end here. This is from Marco, who says, I always wondered how the shell interfered with body temperature control. Is it feasible to consider the shell an adaptation to improve temperature control?
An interesting question. We know, for instance, that osteoderms and crocodilians are important in temperature control, but I haven't seen anything yet in turtle shells that should show the similar kind of vascularization that you see, for instance, in crocodilian osteoderms. When you flip over a crocodilian osteoderm, you see all these little pits, and those are vascular pits. So the osteoderms do, and the overlying structures are involved in thermoregulation, but we don't have anything really equivalent in turtles. Also, turtle shells, the carapace and elements in particular are very sort of spongy, so one could sort of see that they could have had a role in this, but I'm not aware of any physiological studies on that.
All right, I have one more. I don't want to pass up the opportunity to ask you more about cloacal aspiration and how that actually comes to pass. And is it something we see in any other groups? I only know about it at this, that it occurs, but I don't know anything more. To what degree are turtles able to do this? Can they survive on this for a long period of time?
Apparently they can survive on this for quite a while, but it's only an adaptation that has been demonstrated for some freshwater turtles, and I don't think it's sort of something that is used in general. Turtles have come up with a generally very unique breathing apparatus, because once you have boxed yourself completely in, you obviously can no longer rely on coastal aspiration. For an animal as large as a turtle, Euler respiration, that's, for instance, small squamates use, is not an option either. So they developed a system where basically several sets of muscles at the front and at the back of the shell were recruited to change the volume of the space around the lungs and drive one set to inhale and one set, by compressing it, to exhale. So they had to develop a very sort of peculiar system, which is unique among vertebrates. But this cloacal respiration was a real surprise. As you can imagine, it got a lot of public attraction. It proves that assholes can breathe.
Yeah, right. And it just seems like one more way that turtles have developed an ability to survive hard times.
Yeah.
And
I mean, we find all these amazing things. I mean, here in North Carolina, they have found turtles and alligators getting completely frozen into ice and maintaining very, very low levels of metabolic activity. And then once the ice melts, they're just up and about. And we always thought that when it gets too cold, an ectotherm just dies, but they don't do that actually.
Well, we are just about at the time to wrap up, but I have one question that I'd like to ask. As you have demonstrated in your talk, and on it, you've touched on only a part of the story of turtle evolution. There's so many mysteries in the evolutionary history of turtles. What kind of turtle would you most like to find? What would be your number one wish of a fossil turtle to find to help sort out these mysteries?
Well, actually, the number one interest would be to have something that is close to that common ancestor of cryptodurans and pleurodurans. What does the neck of that animal look like? So that would be my, somewhere in the mid or maybe early late Jurassic, that would be a really interesting thing to see.
Because we really don't have that. We have really well-developed pleurodire condition and really well-developed cryptodire condition.
Yeah. But like what would an intermediate look like?
What do those early, like the Triassic turtles, look like? Do they look like more like one or the other? No,
actually, they don't have any signs of a retractable neck anyway. There had been a study recently, it was suggested that Proganocheilus could perhaps sort of move its neck a little bit to the side. But the problem is in Proganocheilus, it had this massive armor on the back of its neck. So that would have really restricted what it could do with its neck. It wasn't very flexible, and it certainly couldn't have pulled it into the shell or under the shell with those structures. There are these very complex osteoderms. They kind of look like the scutes on the back of Godzilla. There's all these little Christmas tree-like structures. And they had that both on their neck and on their tail. And the only other turtles that ever had such armor were the myolanids in the Pleistocene of Australia, which were also very heavily armed.
Very interesting. Well, thank you very much.
You're very welcome.
All right, we'll go ahead and wrap up there for the show. Thank you again, Hans, so much for joining us. We learned a lot.
Thank you so much for inviting me again, and I hope you found this interesting.
Great. And thanks to everyone for watching and for submitting questions. We'll be back for the next episode, episode 60 of Paleo Talks, coming up soon.